This is ISH Q7 introduction part. There are total 20 sections. In this video, we are covering section 1, that is introduction. Section 1, introduction. 1.1, objective. This document or guide is intended to provide guidance regarding good manufacturing practice, GMP, for the manufacturing of active pharmaceutical ingredients, APIs, under an appropriate system for managing quality. It is also intended to help ensure that APIs meet the requirements for quality and purity that they purport are represented to possess. In this guide, quote unquote manufacturing is defined to include all operations of receipt of materials, production, packaging, repackaging, labeling, relabeling, quality control, release, storage, and distribution of APIs and the related controls. In this guide, the term should indicates, quote unquote, should indicates recommendations that are expected to apply unless shown to be inapplicable or replaced by an alternative demonstrated to provide at least an equivalent level of quality assurance. For the purpose of this guide, the terms current good manufacturing practices and good manufacturing practices are equivalent. The guide as a whole does not cover safety aspects for the personnel engaged in the manufacture, nor aspects of protection of the environment. These controls are inherent responsibilities of the manufacturer and are governed by national laws. This guide is not intended to define registration/filing requirements or modify pharmacopoeia requirements. This guide does not affect the ability of the responsible regulatory agency to establish specific registration/filing requirements regarding APIs within the context of marketing or marketing authorizations or drug applications. All the commitments in registration/filing documents must be met. Subsection 1.2, Regulatory applicability. Within the world community, materials may vary as to the legal classification as an API. When a material is classified as an API in the region or country in which it is manufactured or used in a drug product, it should be manufactured according to this guide. Subsection 1.3, Scope. This is the very big part and very important also. Section 1.3, Scope. This guide applies to the manufacture of APIs for use in human drug or medicinal products. It applies to the manufacture of sterile APIs only up to the point immediately prior to the APIs being rendered sterile. The sterilization and aseptic processing of sterile APIs are not covered by this guidance, but should be performed in accordance with GMP guidelines for drug medicinal products as defined by local authorities. This guide covers APIs that are manufactured by chemical synthesis, extraction, cell culture slash fermentation, by recovery from natural sources, or by any combination of these processes. Specific guidance for APIs manufactured by cell culture or fermentation is described in section 18. This guide excludes all vaccines, whole cells, whole blood and plasma derivatives, plasma fractionation, and the gene therapy APIs. However, it does include APIs that are produced using blood or plasma as raw materials. Note that cell substrates, mammalian, plants, insect, or microbial cells, tissue, are animal sources, including transgenic animals, and early process steps that may be subjected to GMP but are not covered by this guide. In addition, the guide does not apply to medical gases, bulk packed drugs, medicinal products, and manufacturing/control aspects specific to radiopharmaceuticals. Section 19 contains guidance that only applies to the manufacture of APIs used in the production of drug or medicinal products, specifically for clinical trials, investigational medicinal products. An API starting material is a raw material, intermediate, or an API that is used in the production of an API and that is incorporated as a significant structural fragment into the structure of the API. An API starting material can be an article of commerce, a material purchased from one or more suppliers under contract or commercial agreement, or produced in-house. API starting materials normally have defined chemical properties and structure. The company should designate and document the rationale for the point at which production of the API begins. For synthetic processes, this is known as the point at which API starting materials are entered into the process. For other processes, example fermentation, extraction, purification, etc., this rationale should be established on a case by case basis. Table 1 gives guidance on the point at which the API starting material is normally introduced into the process. From this point on, appropriate GMP as defined in this guide should be applied to these intermediate and RR API manufacturing steps. This would include the validation of critical process steps determined to impact the quality of the API. However, it should be noted that the fact that a company chooses to validate a process step does not necessarily define that step as critical. The guidance in this document would normally be applied to the steps shown in gray in table 1. It does not apply that all steps shown should be completed. The stringency in GMP, the stringency of GMP in API manufacturing should increase as the process proceeds from early API steps to final steps, purification and packaging. Physical processing of APIs such as granulation, coating, or physical manipulation of particle size, example milling, micronizing, should be conducted at least to the standard of this guide. This GMP guide does not apply to steps prior to the introduction of the defined API starting material.